heard a lot of really fascinating and kind of tour de force of some, I guess, basic science. And I guess what we're trying to do in the industry is apply some of those principles. So the challenge of the industry, we've heard everyone, I think, has got 3 or 4 slides on it, and I have as well, so I will jump over them fairly quickly. I guess I'm going to first off talk about what typically clinical pharmacology folk have been doing in the industry, and then some strategies and how we're trying to solve our exploratory development problems. And then I guess the thing which I guess people who know us at Pfizer well will expect of me is to start talking about quantitative clinical pharmacology. I guess this is our philosophy as to how we can become more efficient and make more appropriate decisions particularly in the exploratory space, but also in late-stage development as well. So this is my slide that speaks to our issues, and everyone's got a similar slide, but yeah, we're trying to get to success, and there's a great big speed bump in the way, which is our inability to turn good ideas into positive proof of concepts. And here's the slide, which you all know, so I'll jump over. And again, many papers have been written about how appalling it is in the pharmaceutical industry and how we're all failing and it's awful and woe is us, and we might as well just give up now. Or maybe not. So, what can clinical pharmacology do to help the problems of large costs and reducing rewards, I guess, in terms of positive studies and leading to successful filings and then successful products? So what do we used to do? And I think we still do a lot of this, and this is not to say it's not important, but it's some of the traditional things we do in clinical pharmacology. And there are a whole bunch of CROs out there who are very expert at doing this kind of stuff. Phase 1 study design execution, we're very good at, in the linear process that the Baroness talked about, moving things through standard phase 1 design. So we do typically safety, PK, and hopefully, wherever possible, some sort of kind of pharmacodynamic markers in first-in-man studies, multiple dose studies. We get interested in biopharmaceutics because we want to have a formulation that actually delivers drug in an appropriate fashion. So we do relative bioavailability, bioequivalence studies. Drug interactions is something I've spent a lot of time in my career working on. That's really important from a safety point of view. We know that LD patients have perhaps different, either particularly PK, but also PD responses to drugs. There are extrinsic factors like changes in renal function, hepatic function, which impact on how we use our drugs. And we're kind of good at this stuff, and clinical kineticists, and I guess I was a kineticist originally in my formative years, we do kind of pretty simple non-compartmental PK analysis, and we can make lots of really relatively useful conclusions based on that analysis. We move into the sort of more technical areas of population pharmacokinetics, which is a bit harder, and you need some horrible bits of software like non-mem, which are kind of hard to use and make your head hurt. But anyway, we can make, again, useful conclusions. And ultimately, if you think about it, any one of the labels of any of the products that any of you guys have worked on, probably 40–50% of what's written on particularly on the USPI, is clinical pharmacology. So we have a whole great business of kind of running so-called clin pharm studies. Now, again, I have to hold my hands up here. I'm speaking as a clinical pharmacologist without a medical training, and I think for those of you in the room who have a medical training, at Pfizer, where the clinical pharmacologists from a medical background tend to work is more in the kind of experimental medicine, translational medicine, the kind of the clinical domains. And we're, in clin pharm at Pfizer, we focus on the quantitative elements. But clearly this is important, and then we've talked about biomarkers yesterday, and again, I'm sure we'll talk about them later on, and actually, having some kind of target mechanism or outcome biomarker is really important, because often we're flying blind if we don't have any kind of translational biomarker. And when we fly blind, we kind of tend to fly into hills and things, and that's part of the reason, at least, that we haven't been as successful as we'd like to be. So, the problem with all this stuff, it's very useful and it gets onto labels and it helps us make decisions moving forward, but in terms of understanding, for example, PK in and of itself, despite my kind of background and training, I trained with Malcolm Rowland and others in Manchester, where pharmacokinetics is really quite important, because that's what they're interested in. But actually, most other people aren't that interested in pharmacokinetics, unfortunately. I was the unusual child or young adult in my school of pharmacy, because I was interested in PK. Every other pharmacist wants to get out there as quickly as possible. But it helps us, okay, understand some of the exploratory development issues, but it doesn't always help us understand why we have no efficacy, no inadequate safety, or no therapeutic index. And the real issue is actually this thing, which again is going to be addressed later, is about the translational biomarker. And actually we've got some kind of in vivo or possibly even in vitro experiment in a preclinical model. And the guys in the lab think it's wonderful and it's their favourite model, and yet when we put things into the clinic, they don't work, despite those models. So the business of being able to translate biomarkers from a preclinical setting is really tough. And also, I think I mean, given particularly in the UK, the way our physicians are trained, actually, some people who have the right skill set and knowledge to do this kind of lab-to-clinic kind of translation is actually quite hard as well. So it's really hard to get good people to do this. So what can we do about it? So one way is just try and make things cheaper. So we'll go and run our studies in some low-cost centre, and we'll drive down the cost of the studies by negotiating hard with the CROs and all that kind of stuff, so we can make the cost lower. So that means, I guess, we can do more for our dollar, and that's just kind of good business sense, okay? Doesn't actually make us more successful, it just means that we fail at a lower cost, which isn't really a great business model. So let's try and minimize the cost. So let's only do what we need to do to get to the next decision point. So we're not going to do 15 drug interactions straight after first in man, because frankly, we probably don't need them. We're not going to have 5 different formulations we're going to explore before we've actually got proof of concept. So we can kind of manage our spend. We're not going to do a thorough QT study before phase 3, despite FDA's wish that we might. We'll try and manage that risk. So we'll only spend money on the things that will help us make decisions. And we can do all sorts of clever stuff like minimise the number of tox programs we run, and therefore the amount of bulk we need to make. And we can kind of make it cheaper. And we can do all the normal project management stuff. We can have buy-ups and buy-downs. If there's a bit of spare cash at the end of the year, we'll knock up another study in the programme. So this is all good, but it kind of just makes things cheaper. It doesn't make things more successful. And I think I've just kind of said this, but I think Pfizer, amongst other companies, but I think we will admit to have tried to do this. Our experiment over the last 10 years has been the more compounds we put in the clinic, the more likely you are to find one that succeeds, because we've put more compounds in the clinic. But I was fortunate. I got asked to go to some kind of teen jolly at Twickenham a couple of years ago, and at half-time they had a competition. You could win a car, a BMW or something nice, if you could kick the rugby ball from the halfway line over the posts, okay, which is about 50 metres. It's quite hard to do that, even though Johnny Wilkinson makes it look easy. And they just got 5, 10 people out of the crowd and said, here's the rugby ball, kick it over those posts. And basically what everyone did, they all lined up and the ball went do-do-do-do-do. And no one was ever going to get that ball over the goalposts. So, it's just, sometimes you have to change the way you do things. We can't just make things cheaper. So, clearly, operational efficiency is really important. We should be doing this as a matter of routine, okay? Because things like electronic data capture allow us to turn around our studies a lot quicker, hopefully improves the quality of the data, so we have less kind of like data queries, all that kind of stuff. Project managers like this because we can squeeze the Gantt charts and make things that look like they're going faster. We can do, and people talk about, we can combine all our experiments into one, and that apparently is more efficient. We can send a bunch of studies down to the ethics committee all together as a package rather than one at a time, and then kind of shorten the timelines. And with all electronic data capture and all that kind of stuff, we can make our decisions on dose escalation and managing adverse events a lot quicker, because it's great, at least at Pfizer. We run a lot of our studies in our own units. I can go home today, sit at a computer and see what's happening to the dose that was administered this morning in our unit in Singapore. It's great. And we should be doing this. Operational efficiency is really important. We should be doing that all the time. So that's just, to me, that's business as usual. But going back to the points that the Baroness made about changing dogma, this isn't changing dogma, this is just kind of doing good business. So, let's think about what we're trying to do here. We've got some novel pharmacology, and we think it might work in whatever disease you're interested in. And if you want to kind of think about some kind of mathematical equation, this is quite a simple one, but in my world, we're in research, we're about delivering positive proof of concepts to the organization. Once we get a positive proof of concept, that asset will be taken on by our business units, which are large, enormous organizations who can get phase 2B, phase 3 studies run smoothly out through regulatory into the marketplace, hopefully. So we're all about delivering these positive proof of POCs. That's kind of like phase 2A, phase 2B type decisions, depending on what you're trying to do. So this is what we want to deliver. Well, okay, well, if we want confidence in being able to deliver that, and frankly, the industry has been woeful in being able to deliver these things in the last few years, well, we've got the two things that are going to help us on that. It's got confidence in the compound, and we'll talk about what confidence means in a minute, and then confidence in the target, so like the pharmacological intervention you're trying to make. Now, I work in clinical pharmacology in the PK/PD world, so I'll translate that into meaning I have an understanding of the PK/PD, and an understanding of the rationale of the drug product or the target. And some of this stuff is actually quite basic. When you actually get your what the Americans call the Pharmacology 101 textbook, the basic textbook that we all kind of learned at undergraduate level, presumably. To have a pharmacological effect, well, first thing, you've got to get the drug to the target. You'll be amazed, and I'll show you in a minute, you'll be amazed how many times we don't do that. The Baroness has talked about neuroscience. That's a brilliant case in point. Many drugs fail in neuroscience, not necessarily because the pharmacology doesn't work, because the drug didn't get in the brain. So why did you think it would work if you didn't know the drug was in the brain? Because that's where the target is. Once you get it to the target, it's got to actually bind to the target. And I think we're usually pretty good at that. We can do kind of biological experiments or pharmacological experiments to show that the drug interacts with the receptor or the enzyme, whatever it is you're trying to bind. And then actually, well, having bound to that target, do you get some downstream function, expression of that PD? This is, I can't believe any of you wouldn't kind of write this down and say, yeah, that seems a reasonable thing to do. It's amazing how infrequently that actually is what happens in the pharmaceutical industry. So we, like many companies, I'm sure, did a review of why we're so bad at our job. And lack of efficacy nowadays is actually the main reason things fail. So I think 20 odd years ago, PK was one of the issues. Toxicology, it still kills things, but PK really now isn't actually that big an issue. Most of the time, in many cases, our PK/DM colleagues are pretty good at being able to predict what the PK profile is going to look like. So we lose drugs now, mainly in the clinic, because they just don't demonstrate efficacy. And I'll show you the data, but we had a lot of compounds. We played a numbers game. We thought putting more compounds in the clinic, in the end, will deliver more successful POCs. And we reviewed, of those compounds that either succeeded or failed, particularly the ones that failed, why did they fail? And actually, many of the teams didn't know why they failed. So they didn't see any efficacy, and there's actually no direct measure that the drug was even present at the target. So particularly CNS target, and CNS neuroscience is an important area for Pfizer. And so we terminated projects. We kind of got a negative POC study, but we didn't really know why it had failed. And of course, that's not a great position to be in.